Giovanni, for your introduction. I am Marta, and with this speech, I would like to introduce you in the world of artificial intelligence in cancer, and in particular in the study of genomics and other molecules to provide relevant models for biology. We will have together a short presentation, a short introduction about this world, followed by two main subtopics, which are the main subtopics of my research, which are gene fusion prediction and multi-omics patient profiling in cancer. So first of all, what I would like to start is to introduce you to the world of artificial intelligence in computational biology. When we have molecular biology and artificial intelligence, we always need to remind the fact that many artificial intelligence models take inspiration from biology. If we think about artificial neural network, the models are inspired by neurons. If we think about, for example, convolutional neural networks, they are inspired by the visual cortex in our brain. So as the artificial intelligence models take their inspiration from biology, it is also true that many molecular biology models need artificial intelligence in order to find good findings in the research. The first thing I would like to start is to share with you which are the data I usually use in my research. And this kind of data are omics data. In fact, in molecular biology, we can have plenty of data coming from images to omics and other kind of experimental procedures. I usually work with omics, which refer to various experimental procedures in biology whose names end in omics. So some examples are genomics, when we want to study, for example, DNA, transcriptomics, when we want to study RNA, mRNA, microRNA, or other kind of RNA molecules. Proteomics, if we want to identify proteins, metabolomics, if we want to study metabolites, and so on. All these kind of data have some particular characteristics. These characteristics are related also to their computational issues. In fact, all this data have usually a high dimensionality. It is really common to have about 1 million features for one patient or one sample. And at the same time, we can also have some storage issue. When we work with RNA, one RNA sample can weigh 30 gigabytes. One whole genome sequencing experiment, so an experiment with the DNA, we can reach about 100 gigabytes. So all these properties, high dimensionality and problems in the storage issue, reflects in all the other computational problems we need to solve when we want to extract relevant biological features. So first of all, we need to check the quantity of the data before starting an experimental procedure. The quality of the data we are using, because if we do not have enough high quality data, our findings cannot be so relevant. And at the same time, we need to pay attention to multiple data sources we have, because if we want to analyze a specific biological phenomena, we absolutely need to know which omic is better to use to identify that specific phenomenon. Artificial intelligence in biology is full of examples. Here I want to provide a very short list of some examples that are already relevant and present in genomics and transcriptomics. The idea is that when we have all this kind of data, we need tools to visualize multidimensional data sets, because we want to be sure about the data we have and how we can properly integrate all this kind of data. We need methods to reduce the dimensionality of the data, because if we can have about 1 million features for one sample, we absolutely need to perform some kind of dimensionality reduction technique in order to be able to use practically all this kind of data. And another really relevant field is the field that is in charge of identifying patient survival, treatment, and drug effect. Because if we have, for example, a type of cancer, we want to identify for a patient in which subgroup of cancer he or she will be. For example, if he or she will respond in a good way to the treatment or not, and so on. Also, we need artificial intelligence technique to represent imaging data and clinical records, but many other examples can be done in this field. Today, I would like to focus on the two major activities I performed during my PhD, which reflect two main biological focuses, which are the oncogenic gene fusion prediction and the multi-omics patient profiling in cancer. Starting with the first area is the area of the gene fusion. What is a gene fusion and why should we care about them? A gene fusion event is a biological event in which two genes after a break are juxtaposed together, creating an aberrant product. So mainly after a break, a piece of, for example, gene 1 join a piece of gene 2. This aberrant product can be oncogenic or not, so can be strictly related to cancer or not, depending on the properties of this aberrant product. For example, a very popular known fusion involved in oncogenic processes is the TMPRSS2-ERG fusion. This particular gene fusion is responsible for about 50% of all prostate cancers. And therefore, we can understand why it is relevant to predict which gene fusions can be involved in an oncogenic process and which of them are not so relevant and we can forget about them. So in this way, in the area of identifying oncogenic gene fusions, so gene fusions which produce an oncogenic protein, from gene fusions which do not produce an oncogenic protein, we need artificial intelligence methods. Existing tools, for example, OncoFuse and Pegasus, realize this prediction on this block, protein domain analysis. So mainly they perform some kind of analysis, and the result is the fact that the gene fusion is oncogenic or not oncogenic. However, this approach has some limitations. First of all, we need to reconstruct the gene fusion. We absolutely need to be sure where the break point, so the point in which the two genes are juxtaposed, is placed. And second, we need to perform a prediction of protein domains, because some protein domains are not well defined and we need to predict them. A protein domain is just a small piece inside the protein, each protein. As a consequence of this approach, we need to perform a Fisher extraction in order to identify relevant protein domains, and this is for sure a problem. All the previous tools, in addition, OncoFuse and Pegasus, do not perform an automated retraining of the models. So meaning that this can be a problem when we have new gene fusions and we want to integrate them in our model. The approach I proposed is based on a completely different approach. The idea is that, can we avoid the protein domain analysis? Can we avoid this step? And the answer is most likely yes, because if we use directly the sequence of the gene fusion, so the sequence of letters that are the building blocks of our gene fusions, we can create a bidirectional longer-term memory network model followed by a convolutional neural network model. This particular model is able to capture the relevant characteristics of oncogenic infusions, and therefore we can successfully identify oncogenic infusions from not oncogenic ones, overcoming all the main limitations we had with the previous tools. In this slide, I just want to present you some results of this kind of approach. The algorithm I developed is called deep prior, and as we can see from these two histograms, we can see that deep prior is more able in recognizing oncogenic infusions compared to OncoFuse and Pegasus, the two competitors. So for sure, the idea of using directly at the sequence and not relying on protein domains is really an interesting point in this area. So this was my main contribution, moving from protein domain analysis to the sequence. The other work in which I spent a lot of time in this during my PhD is analyzing multi-omics patient profiles in cancer. In this context, from a biological and computational point of view, we need to remind that we absolutely need to put all the information together in order to infer relevant biological knowledge. And it is very important that we cannot put all the information, everything together without any specific criteria and build up our artificial intelligence model. We absolutely need to understand which omics is better for which application and trying to merge everything together in a smart way. In the literature, a few multi-omic approaches have been proposed. The two most popular ones are SNF, which is an algorithm based on similarity matrices, and the second one is MOFA, which is based on factor analysis. My approach is again based on a slightly different idea, and the idea is to reproduce the sample with some meta-samples, and with these meta-samples trying to infer the main structure of the cancer we are analyzing. In order to do so, the approach I proposed is based on a multi-layer perceptron with a specifically designed loss function, with the aim to represent with a unique matrix, a unique adjacency matrix, the final one, information coming from multiple omics, in this case, microRNA and mRNA. The basic idea is that the samples, the 1,000 samples, about 1,000 samples, are represented with about, in this case, 100 centroids or pseudo-samples. And assigning each sample to the closest pseudo-sample, we can represent our data in a multi-omic way, because with one loss function, we can merge and optimize the information coming from the different omics. In this way, the final matrix we get is a summarized representation of what we have on the different levels and on the different omics. In this slide, I reported some results of this approach. The approach I presented you a few minutes ago is called NGLF, and it is this one on the right. SNF is the one on the left. From this picture, it is clear that both the algorithms have their weak and strength points. For example, SNF is more precise in recognizing this tumor class, CPTAC class, whereas my algorithm is better in recognizing the CPTAC3 healthy class. So of course, this approach still needs other additional improvements, especially in the definition of the loss function. But the idea is really good, because with one loss function, we can model the events that are occurring on different levels. So which is the take-home message from this short presentation? First of all, is that cancer is a complex disease, and we need all the effort to study cancer, to study which alterations are involved in which specific kind of cancer, and absolutely artificial intelligence will increase in the next years because results we get with artificial intelligence significantly outperform results we get without artificial intelligence. And more in detail, in genomics and in multi-omics studies, artificial intelligence for sure will grow up, and these fields will greatly benefit from artificial intelligence models. So I hope this presentation was just to provide an idea on my PhD work. And if you have any questions, just feel free to ask. So in general, thank you for your time, and I'm here. I also provided some references if you want, so again, thank you. And if you have questions, I'm here.
Thank you, Marta, for your talk. Now I will read some questions for you that we received. Okay, the first one is, can you combine existing knowledge of how DNA and cancer work to improve the performance of networks?
I'm not sure to have understood correctly the question. If we can merge information.
Okay, I repeat. Can you combine existing knowledge on how DNA and cancer work to improve the performance of the networks? So yes, the principle of the basis of how DNA and cancer work can be used in some way for improving the performance of networks, the design networks, or not?
Absolutely, yes. So thank you for this question. Absolutely, yes, because when we build artificial intelligence model, usually we add information, all the information we have, we try to incorporate in our model. Because as I said before, we usually work with about 1 million features. Sometimes we have a reduced number of features because, for example, we know that for some cancers we are interested in, I don't know, 3 or 4 genes, 20 genes, 100 genes, and we are lucky. But most of the time we want to perform a discovery project. And since we are interested in a discovery project, we need to take all the information we have in consideration, and most of these information are embedded in the networks. So at different levels, yes. I hope I answer to the question.
You were very complete, clear. Thank you. That's okay. Now there is another question. Okay. How many feature variables does the algorithm require on the average, and how feasible is it to obtain these features in real time?
Usually the number of features depends on the experiment we want to perform. So at the beginning, we always start with all the features we have. So again, about 1 million, or sometimes 20,000, 60,000, or something like that. In general, we cannot obtain results in real time. So we need time to process all the data, because we can reduce the number of features, but this result is not in a real time. But I would also tell something more. Real time in biology, I would say, is not so strictly important as an accurate result, because just to give you an idea, when a biologist performs the DNA extraction, this person takes some hours to process the biological sample and to put it in the machine. And then from the sequencing machine, we still wait, for example, 1 day, 2 days before getting the results and before starting analyzing it. So by definition, real time is not relevant in this kind of application. So for sure, we cannot wait too much time, and we can get relevant results in a reasonable time. But some processes require time, and for sure we need time, and all of us, we prefer to wait a little bit of time, but to have relevant results, because also for the validation process, we need time, and this time is in the order of months before validating a product or a specific gene in a specific cancer, in a specific tumor.
Okay, perfect. Thank you, Marta. I do not find any other questions, so we have completed. Thank you again, Marta, for your very interesting talk.